Everybody had a good break? I was playing badminton and I'm sweating a lot, so I need to cool off. You guys are really good in badminton. I'm not very good. All right, so any questions about the mitotic stage and everything else that we have covered so far? Somebody made the point that before the cell enters mitosis, before it enters the M stage, it goes through the S phase. You have the G1, where the cell grows really large, and everything, all the organelles are replicated.
Perton tampu tinana karchigore sena tapon te chiri do and tapon nana yo big wanta tikanza ko persugures kuron any depth zugures.
After G1, the cell enters what? S phase. What happens in S phase?
Da Perton tampu tigi jelaya and a condugore sena inskela S phase redorwa, Natsuga. Take up DNA Jotrip Chiagram Batelan to Gorwa. Take up Karichungu Yoresa.
Anyone tell us? DNA replicates, right? After S phase, you have the G2, which is a second checkpoint to make sure that the cell is ready and able to go through mitosis. Go ahead.
Then the S phase or what? And the take up Sanga Sukarchigores and DNA the Jotrip Chigures. Nitiki Jelaya. G2 search or G Nuadecaresena, Taperton Nuate Dugora, Perton Nuatina ko, Tatasha Satskindo Chichagoroa, Satsitina Carichigoresena, Taku Mutu, Taku Taponte, Ko Yanshi Tapon, Zupon Chigambalan Drobaina, Kola Yachi Tango Kanza Sandues, Ko Zupon Chila drag Tadigindum to take Kugeta Tagoris.
If the G2 checkpoint does not work, and it stops mitosis from happening, the cell dies. It commits suicide.
The getting a so parton yibanana UBG and a coge Satskido take a taxichi, Lika Machi by Imbina, and a taponte, and a Yanchi tapon, Zuponji Chigarambalan Dugumaris, Mando Bina Karchigores, and a taponte Shidugures.
And here's an important piece of information. If there is a breakdown of the G2 checkpoint, and there is an error and the cell still goes through mitosis, what do we call this?
Ketis Ruangasuge Perton Yibala Yopigi and the Taxig Hatskitela Trolo Shorches or Questiondo Shorche, Lekatata Machibache, Imbainaya, Machi B Caps in Bainaya, Chaponte Mutu de Tandon, Zuponi Chicken Wall, a Droba in Baina, the Karin Jongres.
Does anybody know? We call it cancer, which is abnormal cell growth. You are replicating a cell which should not be replicating.
Tatanda de Koge, Taxig Saski Leka Chigio Marwa, Chigimepana Metu to Kuchagin Royo or Tapon, that Takon Chavi Dua de Ledena Sugan Deneti Chungu Resta, Deneti Jungu Res, the Tenesis Tamba Karsugresena Koge. That loot the matamba, Sunta matamba, Ranjon Mainbagi, Tapon, Chagutu, Granjan Chigorwa, or Tilakugura.
So the G2 checkpoint is very important because if a cell should not be dividing, it stops it from dividing. It stops it from entering mitosis. And it should stop it from entering mitosis.
Tatadage, Parton Yibalo Yopi, Kurangi, Taxig Satsichigorwa, Satsiti Hachike Chambuchagures, Ketchambuchago, but Jinsakares, and a Kate Tapon. Kuchaguchi Maron Wine by na, and a cool cakeores. But the Gula Gugureda, Tapo Chagui, Jim Maronwate, Machiba de Gugures. This Tambaletigi Chelede, and a Parton Yibala Yopigi, and a cook Satsichi or Taxi Satsiti, and a Kutata Zugures.
Good. So, what we're going to do now for the next 30-45 minutes, I think we'll spend 45 minutes on this, an hour if we need to. This may take a little while. You can all work in your groups. We're going to take this drawing, this particular cell, with these chromosomes, and we're going to draw out the whole mitotic stage. Going from, yeah, go ahead.
Tatanda de kapsanga so chichina kamashi chishenga chichina chiso chikiren de la ya ani ruka ngu gane tananga sugi gangla trisha ju thapon sto ga re ta thapon so ro de thapon de shisha de ta ani zupon ni chige ramba de kanza du sermola yes.
So you're going to start with this drawing, large A, small a, and you could do different colors or you could do the same color, it doesn't matter. I'm going to do different colors just to make it easier for us. And then we have large B and small b. We're going to take this drawing, and in the 5 different steps, you're going to draw each mitotic stage, all the way from prophase all the way to anaphase.
Gelage Kapangar Mutidorwa, Rumundi Tistambalaya, the Denalaya, Natsulaya, Tirwa Karsa, Tsekantirwa, Tsekanti, Yakantiko, H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H.H. phone ni chi ge ramba nga yorwa tampo nga chun dunguni goch ge ta thama thumngu partarwa ti ge te ane remuti trugu yes ane de kap se tribi kap se wa tanda yakan de so gelai bana chonta mando pe cha sa dwa te che so chi thong de che te che so chonta chiba pe cha tana digre chonta mando pe cha tana digres.
So your notebook, you're going to make 5 drawings. What's the first one?
Ta che so ritep gang la ya trugu yo res ramba tampo te ka res
Prophase,
profes
prometaphase,
metaphase
metaphase, anaphase, telophase. Next to it, you can draw cytokinesis to show the 2 different cells, okay? But just remember, it is not part of mitosis.
Aha da da de la ya nga so ge zu phon ni chi ge ramba nga chi ro tingu che tamngu che te ge jilan dungu che jingu che thumngu che tu smati ani ti ge je la ya ani sur to la ya che chu nga so thapon tapthin chi ro thapon tri ge ramba chi ro o ti tna digi yo res imbai na ya thapon tapthin de zu phon ni chi ge ramba na chi ge yo ma res ting ge gur das.
I'm going to give you a big hint. This is where many of my students make mistakes when they do this activity. They forget to remember that the cell has to go through the S phase. The cell has to go through S phase. What happens in S phase?
Ta da ding is nyenda chi tang ge yes ko ge phila yo pil hotu chu ge de ka sa notiti yanse zo go dos ro che chule nyenda tang de ka res stambala ya Chaponti S phase gro S phase te dirwada nga chu ge DNA jotrub ge rambatere do ti jugu res ta te kap se kari chungu yores.
You have to show that the cell is going through S phase through DNA replication. Otherwise, the mitosis drawings do not work out.
Ta tanda de kap se che chu ge DNA jotrub ge rambate trugu yores ti matri ba imba na ti ge thapon chi orwa Digi Tapondi, and Dina Duayo Maris, Karsa, Zupon Chigarambala Duayo Maris.
So essentially, I'm giving you a big hint. You have to replicate that DNA.
Tati Dodo Karsagores and a Gala Tushaju DNA Chuarwa, DNA thing as Sugeta, replicate that Sambala ko Pershizugu Yoris.
Okay? Okay, with your groups, you guys have 45 minutes, an hour. We'll work on it. We'll walk around, we'll guide you. Be creative. I know you're all artists, and I love the way you all draw. Make them beautiful. And at the very end, maybe we'll have some groups come up and do their drawings and show us what they have.
Do they need to work on as a group or individual?
They do it individually, but they can help each other in the group.
Okay.
Everybody does their own drawing. Yeah.
Susu remote roches, susu remote gure, and a young passer ropa go by na yan sit with the chi ropachina de gores, ne danda nga so kama shenga no chido which paro telaya dus remote triens ne tigi jelaya ane mi khase chi ya lang de to susu kari trigo mete gala dus tenchi nana de gores ji yes ti ko
Normally this would be a 10-point assignment and the average is about 7 points or so. It's not an easy assignment. So let's see how you all do.
Ta shirge imbaina dita do ge nangli do chi tre chi gorwa ti te ba imbaina de la ya chwa di tang bi kap se de la ya kama chus tus nang yes chata imbaina ta de this kali khapu do yores ta ko chanyo imbaina ne kama jug na ne duns ma shira ga mares.
Okay? And at the very end, after we do this activity, we'll do our jigsaw, where we're going to work all in drawing different mathematic stages. And we do that at the very end in the next session.
Ta de je la ya nga su ge ludri ki je gu chi ya chia chi yes ta de chi chi na ga su ge chotan de ge ju to ala che che ne de je la ning kong ge chotan chi goro wa de ge smith de na is ne tan da che su ge tan da ge je gu chi do wa trimu tre chi do de tro chis.
Good. Let's get together and start drawing. So we'll continue in about 2 or so minutes. You should be able to finish your drawings by then, and we'll compare to what the drawings should look like on an actual activity.
Kama nyusum chi la ya metu chi tsaba chi ro chis en tik je la ya su su trisha ju de ani ngone ki di kana do chi yo gu sare se ti ta nyam tu ne drais.
All right. Everybody good?
Yes, sir.
Did you like this activity? Yes. Did it make you suffer a little bit? Yes. So even my students at Emory, this is not an easy assignment, and they stumble upon the details on what it means to replicate the chromosomes. But once you understand what's happening, it actually becomes quite easy to finish the activity.
De chirge ko le lauchi yo mares this nyota sauchi ores phila yo pilop to simbana ani ko kasangonjong do theg do sta tanda de kap singe gwati ga so ge chwiz de ko ngoshi zwa jgorwa ti ha ko imbaina ti chi go ge netan de ha ko imbaina ani ko ndo tan de this le lau ro chag yores.
So if we know that this cell, or the one that I drew here, is in G1, well, we know that after G1 we have S, and in S the DNA replicates.
Ta tanda trisha ju ge thaponte desi jabana shre ko parton tampo ge rambatela yores parton tampo di ge rambatela yo paine tigi jela karchi gore S face go S na do gorwa dige nane karchi gore S tambala ya DNA ngushi ju yores.
And once we know that detail, it becomes really easy to finish our key. And this is the actual key that I give my students at Emory.
Da de ko ge lenti tabdu kana do chi yemen de ko phini lop thila tre de ins te ke ti tambu de DNA ti ko ngushi jugu yo pate ne ta ha ko sona ko rum taran tshana ko res ro rum de ne phila yo lop thula chi ta picha sa chi pilansa do ngula ko thu chi yes.
So you go from having non-replicated DNA to replicated chromosomes, 2 sister chromatids joined together, 2 large A's, 2 small A's, 2 large B's, 2 small B's.
Da tanda de la ya pretechu ti sha tangpu te sha ju ka pon ti na la ya ko chekam masna ya chi ma shi ta me do ra ti tambala ya chekam ya chi te ane ko pershi chi bala tene drashi su bala tene de te nga sula ya ane karsa na chong ge ya kan de nyiri nyam te legi yorwa te legi yores ta de le ba na nga super nga ta ba na H1 ni che H1 ni che B1 ni che B1 ni du chak yores.
And then from there on, it becomes really simple. In prometaphase, the nuclear envelope disappears. You have the movement of the spindle fiber apparatus at both poles of the cell. You start to get an essence of lining up, although they're not completely lined up yet. They're still somewhat sparse.
Tanda de kas ni batila ya tat dungu kas te chenyo tamngu kas imbaina ane nga suge thapong ning ko chi ro ati kale kale me per cha bata ane nga suge phangsu ti tribarwa phangsu ti kur dus tripa ane temse nga suge chüzu yo ro chüzu ti imbaina ko pando chi du yo de imbaina chi kimala pa ya ta pa du yo ma imbaina ko chi zuftan do chi ton chi pando du yo res.
In metaphase, what do we call this?
aha da tanda nga su gu dungu la imbai na dungu ka kapsa ku ge kima ge zuta tila galagre.
Anyone? The metaphase plate. We have a single linear spacing where they're completely aligned, and the spindle fibers are about to diverge them, they're about to separate them. And in anaphase, we have the complete separation of the sister chromatids.
Aha da di kapsa imbai na di dungu tigi kapsa tigi mila yan dudar chin dudar ti kapsa imbai na nga su ge tirwa da suizi kanza ani tiwa la ya ku tachi ge tichi gang la ya pando dik de yo res ro ni tigi je la ya nga su jingu ge kapsa karchi go res na ani nga su ge yakang ro na chong yakang ti ani ta phangsi ge pha ten de tang ani ku na chong yakang tu su khate ba chi sha yo res.
And of course, in telophase, we have the emergence of the new nuclear envelope for what will become cell 1 and cell 2, although they're still part of one large mother cell.
Aha da di thumgu kapsa kari chayo re se na ni ningko te ni yang che che to chong de tang ko ni ni la ya ningko ba ni te chayo re sta tandi chala ya ku danda ni kap de la ya ko thapon chembu chi na de orda imbai na ku maung ba la ya ro ta ku ramba jima de la ya ku thapon ni la ya andu sinjun ru yo res.
And of course, cytokinesis would show complete separation of the two cells into two individual yet identical daughter cells.
Aha ta de ge je la ya thapon tripthing ge ramba de la ya kar chi go re se na ani thap na chong thapon ani te ba kan sila na chong thapon tin cha ya chi pa chi chan ni te go yo res.
Okay? Any questions?
Draw yes.
So hopefully you enjoy the activity, and it's a little bit difficult, but it's okay. That's how we learn. It's good to stumble upon obstacles. It's good to question yourself. It's good to ask for help. That's how we learn.
Aha da tanda chi su remote drag chi che ku te tu chong bi ro yes yan san samala ya kange do the hama kon ro china ya go yo res. Da su la chua re ani su tsansama kange tin la dong thi ja re san sama ya rokpa sala ke che tri chi tin ro che ba na ne te la te ne de nga su ya chi lob jong chi ya te yong yo res.
Good. So now, so this is actually a little bit off, but we're going to pretend that we are in the second session now. We're going to talk a little bit before we go into our break, into our lunch. We're going to talk about the method that we're going to be applying for today.
Aha da di kapsa nga so zu tanda go tsu ani ti ge jela nyong ko jabla muthe yes ta tanda di kapsa nga so ge jegu kari chi ge drig ye me ti ko de shi yes.
How many of you have used the method or have learned about the method called jigsaw?
Aha da nga so ludrik jegu chi go ro di ko la ya mi ka su chi ge jang bes.
Perfect, a lot of you.
Yeah, we covered that in the pedagogy sessions, yeah.
Better? Yes. Excellent. So we're going to implement our theme, our active learning theme called Jigsaw. So I'm glad that all of you or most of you already have seen this and have learned. So we're going to focus on breaking the class into groups, which we already have. Perfect. We're all in different tables. Each table will be a different group.
Aha. Da di ludrik chegu di man chu sing yo res ta nga su kar chi gin se na di zendra te ne ko man la gucha ja ge sta da dus ta ko ma tro ko tambo gucha ja se do nga su roxi re te ro re chi gin.
So as you have already learned, Jigsaw technique is a method, a tool that promotes learning in groups, improves student motivation, and learning experience. So you all have a different role in the project, and you're going to share your information and learn from each other.
Aha ta da ge ludrik chegu de ten de dong ko lob ju chi ati kar chi go re se sta bala ya rukho kanang khu ne ani ku nyam nyong tang chi ya zu nyam te pansla ngoto chi bal te ni chi yo re sta de ga ne rukho chi kana ne rum re la ya ani chegu tang chele te manda te ne nyam te re chi wala te ne de te ani kang chi se ta chegu te cha chong wa dre chi go ro ti number te go res.
And this works extremely well anytime you're teaching this particular subject or any subject in science, and you have a large student body. There are 60 of you, give or take. this technique works extremely well on a large group.
다 단다 되게 루드기 제고치거와 루드기 제고데 단다가 조시데레 디난싱게 아니 천리 던데비 아니 그 조시 셴다 디나요 그 페슈동고 페슈동나 그 야부용그레 디난싱게 하버도 아니 루드디스 마요 바이나 단다 데랑아 솔루트 참아던 바이네 두치마스 잠스거와 아티스 담발라야 루드 마위 값스 아니 단다 루드기 제고데 페슈동 바이바나 루드디 차츠부용그레스.
So anytime you're teaching a complicated subject like mitosis, meiosis, cell theory, anything, and you have a large group, I welcome you to implement Jigsaw.
다 지속에 단다들라 영화 속에 주폰 데 니치데 요로와 되게 제라야 아니가 속에 제폰 시지게 넘버 테오리 디난싱게 타풍가 남샬라 소바게 아니 지금 야그 구아칼리 카포 조타 사우요 주고 조시 디나라고 임바이나 아니 그 루칼라 고샤자지 네 루드기 제군디 또 냠트 티바 임바이나 아니 그 릴하와다 아니 차츠 보용 거레스.
What are the steps for Jigsaw? Well, we are making student groups, which we already have. There are 9 tables, there are 9 groups in our class here today.
다 단다데가 루디 제국의 고름 땅부디카레스나 루카즈고요레스 다단가스델라야 루카구두스.
You're going to pick among your groups one student who is going to serve as the leader of the group. We're going to divide the topic of learning into segments. There are 5 steps in mitosis. Each group is going to focus on only one step of mitosis.
다 데나라야 거기 장야기 조시데다 tumbu zu고요레스 다 데캅스 임바나 아니 루카레레레게 단다 주폰 니치게 럼바마 nga요로 아티게나니 칸치글라야 두노치그레스 칸치고야 부장구레스.
So when we come back after lunch, each group is going to get a different number, 1 through 5, and each number will represent a different stage of mitosis.
단다 연공 잡라 치수 초용 스탄발라 가르치그레스나 아니 딘슨 루카레레레라야 아니 안탕 만도라그레스 안탕 시그네가 버트 만도라그레 안탕 티게 가리 총그레스나 아니 주폰 니치게난게 럼바리리 총고요레스.
And each table is going to make a large scale drawing, and I'm talking large, we have large paper in the back. You're going to create one large scale drawing of your step of mitosis.
다 단다 가르치그레스나 로카수수수 응원에 아니 다 쇼고가수 코트치겐스 달라야 쇼고 첨부 된다 요레스 쇼고 첨부 대강라야 수수라 테샤주게 럼바치고로 아주 폰치게 럼바데 아니 그 쇼고 첨부 대강 르무 첨부지 주구레스.
So you have to be creative, you have to be artists, and you have to impress us with your beautiful drawings.
다 데카스 지제고데 드비카스 르무 닌지보트지 길라코스 예원 토바치알라야 니 차시당지 르무데 드구요레스 데카스 드지담발라야 다카스고레 다 크리에이티브 시스템발라야 지 리포트 정보당 지 락세요 파나데 데야 드고레스.
After a few minutes, 10-15 minutes, you create your drawing. You're going to walk around and you're going to find the other 4 steps of mitosis and you're going to put them together for the whole mitotic cycle.
다다다 되게 제라야 다 까마충원 유시치그린라야 수수게 르무티트 산우게 제라 아니 르무티트 산우게 제라 카치그레스나 수수다 파츠도데데 다 수수라야 름바식마시오 마르와 름바식마시메자 네 셴다 담란도데다 름바 셴다 시데 제데다 제누게 제라야 아니가 수수폰 니치게 름바 차 청와데 르무다 앙아요로다 르몽아우티두 스포넘치 드구레스.
And once you complete this cycle, you're going to give us a 2-minute presentation on step 1 through step 5.
되게 제라 가르치고레스 스탄발라야 아니 다 담시 지구 프레젠테이션 젠터 지구요레스 담시 지구요레스 네 그런 당보네 녀스마시와가 버터 간다도 요베당 티콜라야 담시 지구레스.
So now you see that each table represents a jigsaw puzzle combining into the completion of the picture of the assignment.
단다 치수카 통그레스나 루카레레레레레 단다가 수드기 제고는 그 연락 접차레레제 아니 고수 냠든 디스탄발라야 근데 단다 디캅스 아주 주폰이 제게 거기에 넘버 차 청와딘도 스트립트고요레스.
Even though we are not giving this particular step, it's important to also test the students' knowledge on the theme.
다다 딩아 수가 담바디야 제데 지금 말했어 임바이나야 지 롭트가 같이 신도 심도 될라야 네 계제트라도 지지 고수 신도 문도데 아니 체제야 그 전부 차그레스.
So whenever you're teaching, you want to assess your students on their basic knowledge, on how much they learned from what you were trying to convey to them on a particular topic in science.
네, 다주 저런 걸로 선세 트픽업스 아니 티케 전부 차고요레스 저런 걸로 트글라야 티바디 롭트술라 같이 신도 만두르 와 세르기 조셔시 트바치게 트픽업스 쿠츠 같이 신도 문도데야 제시 제케 전부 차고요레스.
Since we have 9 tables, let's make 10 groups. So this table right here, let's divide you 3 and 3, right? So that essentially you have 2 tables in 1 table, okay? So I'm going to give each of you, each table, a number, okay? So on the end, you guys are 1, okay? 2, 3, 4, 5. Remember your numbers, okay? 1, 2, 3. You 3 are 4, and you 3 are 5, okay? So you received your numbers. Remember them for after lunch.
다다 치슬라 장가치라서 하데 삼라 녀데로 치스 녀콘 잡로라야 체구드스 담발에 피치 당고레스.
Each number will correspond to a different mitotic stage.
다 장가 레레레레 치트서 치니 숨싱가 페트트서 데 장가 레레레레 응원에 아니 zupon이 치게 름바 만도리리 총고레스.
So when we come back, we're going to give you large pieces of paper, and you're going to spend 15 to 20 minutes drawing your stage. And I'm talking about a beautiful, artistic, detailed drawing.
다 인권자라 수용 스tambalaya 좀누 제라 까마 중간 유시치나 텔레 수수라 라데 주가. 다 예나 마시 요요 말다. 제수지 생이 세대는 요요 말와. 로 인권자라 존 스tambalaya 수수수라 땅가 같이 라요 바티게 아니 zupon이 지가 럼바티 리모트로체스 리모트리아티 닌제보제 자제보제 아니 코나틀레 시성사고로와 어디는 트구레스.
Be as detailed as possible and do not draw other mitotic stages, only the one that was given to you.
다 트리바데 간 제제 트구레 자제보 트구레스. 데트 고아탄데 티기 제라야 수술라 테샤지도 마세 젠데 트구 마레스 수술라 야름바카리라요 바티 트로 만다 젠데 트구 마레스.
After you finish your drawings, you're going to walk around, find all their 4 stages, and complete the cycle from beginning to end. So we should end up with 2 different complete mitotic stages.
다 티기 제라야 아니 수수 드로데데 아니 름바 젠다 시트요 비기 메테다 냠트 치데데 아니 차랑라야 zupon이 지게 름바 차츠모트 주지 zu구레스 다 단당아 수두스 고고셰 자탕라 테바이바이나 마술라야 르무 차츠모테나도 아니 용고 요레스.
So you're going to spend a few minutes putting them together, and you're going to present, you're going to stand here with the drawings, and you're going to present to us the whole mitotic stage.
다티 트누 제라야 아니 냠트 제다 코 르무가 요치테 루칸가가 냠트 용고르와레 미타 미가스 루칸가 냠트 용고르와 르무가 들어와 틴데 큐온데 탄 들로네 진단 들로네마 르무티 텐션 니 나톤데 시작고레스.
So group with the number 1 will draw metaphase.
네 루카라야 앙가 타보라니 치고르와 제수카레 트구레스 네 둥우 트구레스.
Group 2, telophase.
루카 니바데 안타 니바라니 트주게 담구 트구레스.
And you see they're out of order, right? This is meant to be on purpose. 3, prophase.
somebody did trugures. That is that the two days.
4, anaphase. And 5, prometaphase.
Tanda de Tanka Tita inch de the poeti two days. I hit the Zuchijagorwa. Tamputi did the one as a metaphase, metaphase lanan dungur, Tamputin dungoche. And you are the telephase lana did the telephase lana tumuche. Somebody profess to the profess in Bambana Tungurwa. Inchi telesharoche, poeting is Zuchijagua. Tani Shibati NFS, the NFS, the Anje de Dua, Chingur Dua. Ani Ngabati prometaphase lana tamu. 이 보이티 단다 주시자셔. 야.
Once we go through this cycle, we're going to change the numbers and you're going to repeat it with a different drawing, with a different stage.
다다 디 두스지게스 당보텔라야 아니 지 트리거 와테 지 트리거 와티 찬우고 제라야 이 양셰 양가츠로 거레레레라야 아름바 만다와티테 아테르무트레 내가 수글 제구디 제드디 지드케스.
Good. So don't worry about memorizing these numbers. We will, after lunch, I will open up this slide again so you can see your number and you can see your picture, okay?
데 ngacho tangu mares zenda jela ya de yangche tengi su tashin tachina degrees tada de poiti no de sing yangche sujao.
Good. So now, before we go to lunch in a few minutes, let's spend some time looking at actual mitosis pictures in biology.
Da ngacho person de matengwe la ya ani jingwe ge na ni zupon ni chi ge ran ban du shin be ge per la ya per ta nguni chi gorwa. So
it's very common. For example, yesterday we tried cutting our root onions, right? Our onion roots. And we tried to get some slides to see what they look like. And a professional slide would look something like this, where you have a picture with all kinds of cells. And as you could see, you have all kinds of different stages within this particular group of cells.
The Chitan Bana dinner do your race, right? The day and I got to tapon mampu tomba gansla and tapon chagugirwa, Zupon chigir and banane, Goramba manda, manda, you tapon mampu tongus, dinner.
So anytime in the future when you're teaching classes and you're teaching, for example, cell structures and mitosis, practice looking at pictures like this and test yourself to see whether you can recognize any particular mitotic stage in this particular group.
Jema Chitsukul Thompson Tribi Capsi Bana dinner do picture down that the Susuge ani dinner Zupon Chigarambati dinner like Dumaduti Chitach or wrong Winsent can duty Diati John the China Yagures.
And sometimes, even though we teach mitosis with 5 steps, it's not like in any given cell, those steps are sometimes blurry between each other and it's difficult to see in which stage they are.
Tanda chirgin bananga so de la ya ko ramba ma sona ramba nga na de se jagu yores sam la ya generative caps pandina do in bana yang ko ramba chitan ramba ni ko paru ko pansante ani chi sela masela dina yung gre sela masela on stambala di dire se ramba ti ngunji che khabu ni tan dina yung gre.
Sometimes it becomes really easy. For example, and here's my test to you. What stage is this cell right here?
That handsome la ya ta ti li law yores that pisha cha paina igela chikten so di gares.
Anaphase, right? Easy to tell. You have the separation of the sister chromatids. Easy, right?
That tanda di bana kare chingu res chingu di kaps kare chigidu sena nga chu ya kanti na chu ya kanti su khate du do at istambala di nginji chali law res.
How about this one right here? You have one cell and these two right here. What is this?
Di gare dos.
Telophase, right? So sometimes it becomes really easy. I'm going to give you a tough one. How about, make me proud, this cell right here. If you think this is prophase, raise your hand. If you think it's something else, raise your hand. What do you think it is? Prometaphase, okay. What else?
Yes, someone says interface.
Who said interface? Anybody here agree with interface? So only one student is correct. This is interface.
The interface.
How do we know that this is interface? Anybody? Can you see chromosomes?
No.
Chromosomes are decondensed, right? In mitosis, chromosomes are condensed. So if you cannot see chromosomes, you are not in mitosis, you are in interphase.
아하 다다게 가수 데 주폰이 지거는 바텔라 해용스다 발라 카르치고레 최제티 간자고 척담부 차대로 컨덴스 차대 독부 차대로 아래 말에 이스탄불에 통구요로 다 통구 매바인바나 그게 척들 쇠대요 그래서 독부 차대요 말이스 이스탄불에 디콜 해고요레 척담부 매바데 니 벌굴레고 그래서 인터페이스라
You could have another set of pictures. What do you think this is?
디가레도사
Metaphase. What do you notice? What did I tell you to look for when you're looking at stages of mitosis? The what? Metaphase plate. Right?
데르도 와가 둥거르도 와 둥고 임바나 땅게 셰바나시 둥고 캅스 카리체고레세나 두더세스도 둥고 바로라 카트게 판드게 지도와 티체고레스
What would this be?
디가레도사 Prophets?
Prophase. How do you know? Because you can see chromosomes. They are condensed, so you could see them condensed, but they are not paired. So you're at the beginning stages of mitosis. You are in prophase, and you can still make out the nuclear envelope.
Prophets or
Prophase.
Prophets. 데 땅보텔에 둥울라레스 단다데 가서 임바나 최즈티 두부차데 통트도 와스 로 아니 디가라야 디캅스 디도 가사 그게 닌코지고로 닌코티 갈레칼레 메도아게 네탄데 도와스
Just a couple more before we go to lunch. What would this be?
디가레사
Telophase, right? We have the nuclear envelope of what will become two new cells. What would this be? Anaphase, right? What would, let's see.
단다데 땅보데 가서 데르도 tum거도 와 로, 땅보테데 녀테데 징을 소소 지르도 와
Yes. Interphase, right? Remember, you cannot really make out chromosomes. They are decondensed. This is not mitosis. Interphase.
나하다 단다데 가서 최즈티 통해도 와 최즈 통해 메부강라야 최즈티 고 척담부 차요 말아 두부 차요 말아 디스담블은 디게 네탄델라야 고 고요레스나 벌고나 요레스
And like I said before, cells most of the time are within interphase. It is a very short amount of time that they're going through mitosis.
아하 다치수 가랭이 고레스나 차폰테 고 차폰고 코르징나라야 계시셔봐 임바나 고뒤 페치와라야 고 벌고나 요레스로와 디게는 뒤인 통통 지그rel라션이 고다 디가사 고 차폰 차국은 고 지어와 텔레 요레스 차폰 차국이라나 디갑스즈폰이 지금 고라 요레스
And just like we can have plant cells with a clear cell wall, we can also practice with animal cells.
아하 다다다디 임바나 가수 지싱그 같이 지싱 임바나 고 차폰 고 지무디도와 실로코와 셀월데 디나싱 가수 그냥 소적 차폰라 타나 디그레스
Again, most of the cells are going to be somewhere in interphase where chromosomes are not quite obvious. They may go through anaphase with the divergence of the sister chromatids or somewhere in between within mitosis.
다다데게 소적 차폰데 타바임바나 차폰 만치슈고 벌고라 요레스 다신다 임바나 데나네 같이 타바나 고 가수 징고라 도와 징고라야 갈라나 최강데 수수수라 카트레드야게 남바데 간라 도와
Last two, and then we'll go to lunch.
아하 디가레사
Why is it not interphase?
벌고라 메피게 진생 가리레사
You can see the chromosomes are condensed. You know you're mitosis. You are not in interphase. There is no pairing, right? You can just about see the chromosomes. Prophase.
아하 다디가 둥우레스와 디 둥우 임바다 아니 벌고 멤버 진생 가리레스 담발아야 디 값스 최즈디스 두부차다데 가수 그 자동 치쇼라 통해도 와스
And this one should be easy.
디가레사
Telophase, right? Right at the end of anaphase, beginning of telophase. Very good, my friends.
다 다다디 깔라는 동거리스.
Very good. All right, so let's take our lunch break. We'll come back at 3 p.m. Come ready to do our jigsaw puzzle. We're going to build our pictures. We're going to build our mitosis creations. And we're going to present our results when we come back.
that's Sumbalaya, this is Nabalaya, Nasuke, Zupon Nichige, Pound to the remote no jelayan, Zandana Motocha get the Chebuti dries.
So enjoy your lunch. We'll come back in 3 hours at 3 p.m.
Sumbalan Jayons.
Thank you all.
Hello to Naros.